Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker on the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Andrew Hanneman from Charles River Laboratories.
Thank you, Leah. Welcome, everybody. I appreciate your attendance today. I miss being able to speak in person with everyone, but that'll change soon enough. Let's all hope. So I will be discussing conductions of biologics impurities investigations using mass spectrometry. So I'm with Charles River Laboratories. My background is in analytical chemistry and specifically mass spectrometry. And our facility is located just west of Boston, Shrewsbury, Massachusetts. So the first slide here, just to go over purpose and topic. So again, the purpose is to discuss various types of impurities that will be found in biopharmaceutical products, as well as how to characterize these using mass spectrometry approaches. And then topics, I'll discuss the various classes of impurities and how we would go about conducting an impurities investigation. And then, of course, the most interesting part is to actually get into a few case studies. Okay, so biological mass spectrometry. I've been working in this field for about 15, maybe a little more than 15 years. So I've had an opportunity to see quite a few different types of biologics drugs. And generally, for the most part, they've been large molecule proteins. So of course, monoclonal antibodies, fusion proteins, and then interestingly, more recently, lots of adeno-associated virus products, or AAV. We've also, over the years, worked on quite a few glycoconjugates, so carbohydrate-based drugs or carbohydrate linked to other molecules. We've also worked on lipids and oligonucleotides. Because most of our experience has been in protein analytics, that's what you'll see mostly in the case studies I'll present today are oriented around protein biologics. The picture on the right is from Acadia National Park in Maine, which is a favorite place to vacation in the summertime. So enjoy that. So conducting an investigation. So, really the goal here is essentially to answer the question, what is it? So you have an analysis, you expect to see certain peaks, for example, and then suddenly you have something new that appears. And you need to be able to just figure out what that is in order to control it. And so really what we're talking about here is trying to provide as much information about the chemical structure of that impurity as you possibly can. And really the focus of this talk is to forward the message that mass spectrometry really is the fundamental first technique to apply in these situations. And sometimes we can solve the problem completely using mass spectrometry, but other times we might need to provide additional techniques. So a good example might be nuclear magnetic resonance spectroscopy, or NMR, which can really dial in a little bit more into the underlying structure of a molecule. Okay, so this just shows how Charles River supports analytical using analytical methods to support drug development, and just showing the biologics drug development timeline. Of course, later on in a program, getting to clinical phases and post-market, GMP release methods are needed. So oftentimes these types of methods are going to be a little bit more focused on, say, HPLC methods or capillary electrophoresis, methods that don't necessarily involve mass spectrometry. So typically, people think about mass spectrometry earlier in a program. So as highlighted in green, preliminary characterization studies that will support, proof of concept. We've made this drug. Is it really what we think it is, for example? Or to support IND enabling studies, so fundamental characterization studies that will go into your IND package. And at that point, it's oftentimes advantageous to move away from mass spectrometry just because it's challenging sometimes to use mass spectrometry in a GMP environment. Nevertheless, one of the main points I want to make here is that mass spectrometry really is needed to support these programs all the way through post-market, because you never know when a new peak may arise in a certain analytical method. So you always want to keep that in mind, to have your method so that there's a route to getting new peaks characterized. Okay. All right, so some of the types of impurities that we will be discussing, product-related and process-related. Probably people are familiar with these concepts. So according to ICH Q6B guidelines, and again, some of these are more related to protein and protein drugs. So some of the product-related impurities might include molecular variants, such as truncated forms, different glycoforms, aggregates, precursors, degradation products that might arise during manufacture or storage. So these might include clips, other types of variants, for example, oxidized species, deamidated species. There's quite a wide variety of different chemical modifications that can occur to biomolecules. And then secondly, process-related molecules. So these would be typically small molecules or polymers that arise from the production process, or they might even be molecules that are purposely added to the drug. So maybe like a tween 20 or a tween 80. That molecule itself, the tween, might be modified over time, for example, oxidized or enzymatically clipped, things of that nature. So it's important to be able to provide analytics that can support product-related as well as these process-related impurities. So what we'll talk about really in this talk is mostly going to be focused on primary structure. So mass spectrometry traditionally has been a little bit more focused towards primary structure, so solving the chemical formula, understanding how the different constituents are linked to one another. And so that really is enough information to cover an entire talk. So this would include, as I mentioned above, glycoforms, modified proteins, phosphorylation, PTMs, deamidations, things of that nature that need to be carefully characterized and understood. Now, what I will not really cover today, and in fact, I'm not really an expert in these areas either, but this is a whole other area of mass spectrometry, is higher order structure. Now, we might think traditionally that higher order structure would relate more to biophysical techniques, for example, CD or DSC, DLS, things like that. But I just want to make a note here that mass spectrometry is currently making a lot of progress into higher order structure. And so folks are probably also familiar with ion mobility and HDX, also charge determined mass spectrometry. Again, I won't cover those topics just for sake of time and also expertise. Okay, so this is just a bit of a fundamental slide here describing what mass spectrometry is. Typically, it is used in conjunction with HPLC or UHPLC, ultra high pressure liquid chromatography. The other thing that I'd like to mention is that it's really advantageous to use a dual detection scheme. So that's what's shown on the right-hand slide, where the top trace, this is just showing a glycan profile, but the top trace is a fluorescence chromatogram, and then the bottom trace on the right side in labeled B is the ion chromatogram coming from the mass spectrometer. And you can see how all those peaks kind of line up with one another. So this carried out by just putting a flow splitter in line so that part of the flow goes to the fluorescence detector and part of it goes to the mass spectrometer. And so this is one of the major advantages of LC-MS, is that it can be used with other detectors. So then just to slide back over to the left-hand side, just to talk about how mass spectrometry works, I mentioned that you might have to use a flow splitter because this slide shows that mass spec is what you would call a destructive technique, which means that these molecules, as you can see in the slide there, the sprayer tip nozzle is actually concentrating the eluent from the HPLC into small droplets. These droplets are charged, and then they are introduced into the mass spectrometer, as can be seen on the right-hand side of this graphic on the left. And so at that point, these even very large molecules like intact proteins can be charged and put into the gas phase. Once they're in the gas phase, then they're introduced into the ion optics and other parts of the mass spectrometer, and we'll go into that a little bit in further slides. So mass spectrometry, the things that I will really focus on in this talk that mass spec will provide are accurate mass, which is used to provide a chemical composition, and then MSMS, or tandem mass spectrometry, which fragments the molecule once it's inside the mass spectrometer. And this will give a little more insight into the structure beyond just the chemical composition. And finally, mass spectrometers are also able to separate molecules into the various isotopes, so that there will be an isotope ratio pattern that one can also look at. And this is really useful also to verify the composition and structure of that molecule. And as I mentioned above, sometimes all this information right here can solve an impurities investigation, or it might just be the starting point. Okay, so again, just to get into a few of the case studies, I'll talk about a small molecule, a polymer, intact proteins, peptide maps, and just wanted to mention that these are increasingly being used in a quantitative fashion, also to follow some degradation events like I mentioned earlier, oxidation and deamidation of certain amino acids, and these are coming to be termed multi-attribute or MAM methods. I'll also talk a little bit about HCPs and other peptide impurities. So also just wanted to mention some tips and tricks, and I think that I did kind of touch on this above. Now, maybe it's just because I'm an LC-MS type of a person. I also like to mention to folks that if you choose CE a little bit too early in your program, just keep in mind that CEMS has historically been more challenging to interface with a mass spectrometer, and there are various reasons for that I won't really go into today, but I tend to recommend to develop HPLC methods, maybe initially, just in order to make sure that those methods can be monitored using mass spectrometry. And I also had mentioned dual detection. So again, sending part of the flow to your UV detector and part of it to your mass spectrometer, at least during to support early method development activities. The other thing that I sometimes mention is that HPLC mobile phases can be made in various ways. Keep in mind, if possible, to make them mass spec compatible. And really what that means is that, as I showed in the previous slide, all the components in the HPLC mobile phase should be volatile as possible, so that they can be put into the gas phase and introduced into the mass spectrometer without clogging and making it too dirty. So just a couple of things to mention there as tips and tricks. Okay, so the main workhorse that we will be sort of focused on in this talk are high resolution mass spectrometers, and that'll come more into focus in the next slide. But the key to using high resolution instruments is that they will provide accurate mass analysis. So what we use at Charles River include the thermal orbit trap fusion, and this provides also various fragmentation modes that can be very useful to get structural information. And then it can be interfaced with various UPLC or nano-LC instruments. We also have a static nanosprayer. And of course, UPLCs with various detectors are also quite handy during method development. This just shows a thermo-vanquish with UV, fluorescence, and CAD detectors all built into the same stack. And then on the right, Agilent QTOFs, we use the 6550 and the 45XT. XT itself is really nice. It has an extended mass range, so that can accommodate larger proteins. Okay, so just to kind of dig into these topics a little bit more, first we'll talk about accurate mass. So HRAM, or higher resolution accurate mass, is demonstrated on this slide. And so this is really fundamental stuff here. If you look on the right-hand side of the slide, it's showing carbon 12 versus carbon 13. And what's interesting is that about 1% of all carbon atoms are carbon 13. And so what this does, of course, the same is true for nitrogen and oxygen. Other biomolecule atoms come in various isotope flavors. And so you can see there's an isotope pattern. If you look now to the left of the slide, what this means is that you'll see for various molecules a pattern like that, an envelope, an isotope envelope. And why this is important is that if you have a small enough molecule, for example, 1,000 Daltons or less, a high resolution mass spectrometer will give you a really nice, what they call monoisotopic peak. And that's the one on the left-hand side. So I'm showing angiotensin here. You can see that there's a very sharp peak there. There's 4 decimal points of mass accuracy. That's extremely advantageous for providing a chemical structure. Now, as you move to the center diagram there showing insulin, now this is at almost around 6,000. You can see that the monoisotopic mass, the intensity of that is going down quite a bit. And of course, the reason for that is that now every one of these molecules is going to have carbon 13 and other isotopes. And so it makes it a little bit harder to get an accurate mass. And you can also see how the resolution of the mass spectrometer is also starting to fade a little bit because the molecule itself is quite a bit larger and the instrument is not able to really provide that resolution. And then so on the right-hand side of that same slide shows myoglobin, a protein of 17,000 Daltons. And at that point, you no longer have a monoisotopic ion. And so you're having to use average mass at that point, which is really just looking at the midpoint of the entire isotope envelope. So really what this slide is saying is that oftentimes to solve an impurities problem, you may have to get your protein into smaller pieces. So for example, peptides, and analyze those peptides so that you can obtain high mass accuracy. All right, and this slide here is now showing MSMS, so fragmentation. So it was mentioned above how to introduce ions into a mass spectrometer. And here, this is just showing a collision cell. So on the left-hand side, once you have the biomolecule in the gas phase and ionized, it enters the instrument. This is just showing some ion optics, but then there's some quadrupole filter that can select a certain ion, and then it goes into a collision cell. The collision cell might be filled with nitrogen or helium, something of that nature, an inert gas that will then fragment the molecule. And this is just showing a peptide here. So this peptide is acetylated somewhere on the peptide, and that's all you know at this point. But by fragmenting the peptide, it's possible to get a series of ions here, and these are labeled as B and Y ions, a series of ions that will locate exactly where on that peptide that acetyl group is. And this is an example from an AAV. AAV molecules tend to have acetylation at the N-terminal of VP1 and VP3. And so this is just information that corroborates that that's where that acetyl group lies. Okay, and then finally, as I mentioned above, confirming the molecular formula. So using the isotope envelope. So this is just showing from data from a QTOF on the bottom trace there, the actual observed isotopic pattern. And then the top shows the theoretical isotopic pattern. So once you have a good accurate mass and maybe an MS-MS spectrum, this is one last piece of information to check before you start to feel a lot more confident that you've really solved the chemical formula, at least, is to just check and make sure that these isotope pattern matches the theoretical pattern. One other point that's just kind of mentioned on the right-hand side is that different mass spectrometers may vary in their ability to provide this observed pattern on the bottom. So for example, certain ion traps, if they get overloaded with too many ions, that can kind of distort that pattern. So I just kind of want to keep that in mind in terms of which instrument is being used. All right, so now to just go into a kind of a simple example here. This is an impurity that arose during long-term stability. What you can see on the top left-hand side is that there's an impurity that shows up in this UV chromatogram, and that impurity was then isolated, and that's shown in panel B on the left-hand side. And once that was isolated, we were able to put it on the mass spectrometer. And again, this is a small molecule, so the experimental isotope distribution is quite well defined, and an accurate mass was obtained as shown in panel D on the right-hand side. And that mass provided several possibilities of what the chemical formula might be. And then what became interesting in this case is that the isotope pattern that's shown here, it became obvious that there must be some kind of a chloride containing, it must have been some sort of chloride containing molecule, just because chlorine comes in a ratio of chlorine 35 versus chlorine 37. And so that kind of jumps out from that, looking at that isotope distribution and then comparing it to the theoretical. And then we were able to, as shown in panel C on the lower left-hand side, fragment it and actually assign the fragments as well. And this provided at least preliminary structural conclusion that it was 2,3-dichlorobenzoic acid. And then once we have that information, we can just purchase that chemical directly from a chemical supplier and run that side by side, as the final proof. So in this case, really, this whole project can be run just using mass spectrometry alone. Okay, and now I'll just shift into a similar kind of a discussion around an ADC, an antibody-drug conjugate, just supporting process development. You can see in panel A, this drug is a protein peptide conjugate with a linker, and the reverse phase method showed a series of small impurity peaks, and there's just sort of a zoom view down below in B, showing the total ion chromatogram of all these small impurity peaks. Again, the nice thing about this impurities investigation is that the HPLC method in the traces A and B had been set up, as I mentioned earlier, using volatile mobile phases. So there was no need to do any further HPLC method development. It was really just a matter of taking this method and hooking it up to the mass spectrometer. So once we did that, we were able to get a series of masses for all of those 22 peaks there. and then also to fragment them, and that's shown in D. Of course, this is proprietary data, so I can't really show what the exact protein or peptide sequence is there, but we're able to match that B and Y ion series to the proposed peptide and just show that it was really a degradation product of the main conjugate. All right, so shifting gears a little bit, discussing process residuals, particularly for anyone that's involved in the AAV field now. People know that PEI, or polyethylene imine, is used as a transfection agent to introduce genetic material into AAVs, and it's important to make sure that your process is removing PEI. So this just shows how mass spectrometry is used to help support HPLC method development, so that HPLC method can then be used to monitor various AAV batches for clearance of PEI. So PEI is a polymer, and that's shown on the upper left-hand side there. And I'll make a little plug to Wikipedia. Everyone go out and donate to Wikipedia. So it just shows the structure of PEI with a 43 Dalton base unit there, 2 carbon atoms and nitrogen. And then the method that's used here by HPLC is typically going to involve this sort of step gradients, because what that does is it kind of compresses the polymer into a sharper HPLC peak. So early in method development, it was noticed that this particular HPLC method, although it had some promise, there were extra peaks in the chromatogram that couldn't really be understood unless this method was, again, hooked up to a mass spectrometer. And so what this is showing in the bottom 2 traces are EICs, or extracted ion chromatograms, of 2 different ions that are formed from PEI when it's fragmented. And then just showing that, again, on the lower right-hand side, showing the full mass spectra of those 2 peaks, the 2 impurity peaks. So what we're seeing is that PEI in this particular HPLC method isn't coming out just as one main peak at 10 minutes, but there are various other forms of PEI that are eluting in different regions of the chromatogram. And so this is where it's very useful to be able to quickly hook up your HPLC to a mass spectrometer and check what's going on. So in this case, it was decided to use a different HPLC method because various gradients had been tried to pull all these species together, but really what had to happen was using a different type of HPLC column. Okay, so this slide here is now moving into analysis of intact proteins. Quite a few studies that we support involve using forced degradation conditions in order to understand the degradation pathways of biologic protein. And this is also just used to help support stability studies. So this particular protein was subjected to all the conditions shown in the table on the left-hand side, quite a few different conditions. And then a reverse phase method using UV detection was used to profile all of these various degradation conditions. And those traces are shown on the right-hand side. And so it's shown in quite a zoom view. Even the drug substance itself did have small amounts of various impurities in it. And so the goal here is to just characterize what all those peaks are. What are all those impurities in the various chromatograms? And again, the way to solve this is to just have an HPLC method that's fully mass spectrometry compatible, so that it can simply be then run into both a UV detector as well as the mass spectrometer. Now, once we start talking about proteins, of course, we're moving away from working with a monoisotopic mass and really now using average mass. So this particular protein is up around 17,000 Daltons. And so we're not really using all the decimal points here anymore to say exactly, 17,000.6693 Daltons or anything like that. But nevertheless, these peaks can still be characterized on the basis of some known types of reaction chemistry that happens to proteins or various amino acids. So in this case, I just am pointing out this one peak here, peak 7. So proteins often come with deamidation sites. So these are asparagine, glycine, or NG sites. These are particularly susceptible to deamidation, where asparagine is converted to aspartic acid, and there's a cyclic intermediate that's observed quite often. It's at minus 17 Daltons. And so this is just showing that the bottom trace on the right-hand side is peak 7, which is being assigned as the cyclic intermediate of a deamidation peak. Some of these other peaks then were also assigned as deamidations and oxidations. Okay, so probably quite a few people are involved in the AAV field. So this is just showing some more intact protein analysis related to AAV. Sometimes AAV gels, for example, or CE traces will show, for example, an extra band. And this is one thing I'll just focus on in this slide quickly here is on the lower right-hand side, this VP3 truncation variant, which is quite often observed. And so this is where the VP3, there's a variant that starts at a different amino acid. And this is, and again, another example of where LCMS is quite useful. So you can directly get a readout on the presence of this species. For example, if there is this low abundance band that's present in a gel or a CE, for example, CE trace, it can be quickly assigned using mass spectrometry. This slide also just shows some other variants that can be observed in AAV proteins. So VP2 often is not observed with acetylation. This just shows an example of where two species are present of VP2, one with acetylmethionine, and this results in a plus 172 Dalton mass shift there. And then these aren't necessarily considered impurities. Just on the top right-hand trace, phosphorylations will often be observed using this method. Okay, so to go a little bit further into characterization of AAVs, the cartoon there shows a binding face of an AAV. A little bit of a typo there. It says capsid. It should say capsid, but this is an AAV capsid. So AAV carries single-stranded DNA genetic material inside of it, and it's important that this binding face of the AAV is not too heavily modified so that it's able to bind to the correct receptor, the AAV receptor. And so this is one of the reasons why these VP proteins need to be monitored. And to do this, we use peptide mapping. And again, the point of this is to generate peptides from those proteins. Now, all these peptides are small enough where we can obtain a monoisotopic mass for each one of them and assign each of those peptides to the overall VP protein sequence. So on the upper right-hand side is shown a peptide map with all the peptides assigned, and then lower left-hand just shows the different regions of that full chromatogram and all the peptides assigned. For the purposes of this talk, one of the ways of using this information now then is to track these deamidation sites, oxidation sites, because if those sites are present in this AAV binding face, it's good to know about these. They may need to be engineered out. For example, if there's too many NG sites, something like that, on the binding face, it'll change the charge dynamics. Deamidation will introduce a negative charge. And so these peptide map methods are increasingly starting to be used more quantitatively. Traditionally, peptide mapping was not as much attention was paid to using them quantitatively, but this is changing now. There's a reference here on the lower right-hand side about multi-attribute methods. If anyone's interested in that, have a look at that. But it's a way of using peptide maps now as more quantitatively. So as mentioned, for example, to track the level of deamidation or the level of oxidation to specific methionine or asparagine residues on the VP proteins. And then this is just kind of carrying that concept just a little bit further. I mentioned this earlier in the talk, using MSMS to precisely locate modification sites. This slide just shows the assignment of a deamidation site to a specific region of a VP protein. This is just showing one peptide, and the bottom of the trace here just shows the EIC or extracted ion chromatograms that show where the sort of the main peak elutes in the chromatogram versus the modified version of that peptide, which is shown in yellow. And then these are the MS spectra that just compare the two peptides. And without going into too much detail, it's just by looking at this type of fragmentation pattern, you can see, you can clearly locate which one of those residues has been modified. And of course, it's the asparagine, which has been converted to aspartic acid, or D, in that middle trace. Another consideration around AAV, it's often the question comes up if AAV is glycosylated. One way of tracking glycosylation is, again, to look at the peptide maps carefully using, these are called oxonium ion plots. What these are is a series of extracted ion chromatograms that are showing different fragment masses of sugars or sugar rings. And in this example, you can see there really is no data. So this is really showing that this AAV does not have glycosylation. And if there is any glycosylation present, we typically will start to think about host cell proteins. So another way of using these oxonium ion plots is to get some indication if there may be a significant amount of background HCP present in a particular sample. One of the issues with AAV now is that the purification strategies may not have been quite as fully worked out. Again, I'm not an expert in this area, so I don't want to say too much. I know that with traditionally monoclonal antibodies can be cleaned up quite well using, for example, a protein A capture step. So you can minimize HCP quite a bit. So it's one thing to keep in mind when developing AAV products is the potential for background HCPs, and these types of plots can help distinguish whether or not these types of impurities might be present, because typically AAV themselves, we're not seeing them to be glycosylated. And so just to dive into this concept or the area of HCP just a little bit more, this is a busy slide, so I'll try not to go through all of it. Really, the main point of it is on the lower right-hand side is just to obtain a really comprehensive risk assessment of HCP in your process development. So traditionally, ELISAs have been used, and so that's kind of shown on the upper right-hand side, application of an ELISA. And what we're showing there is 2D gels, one with a silver stain, another showing Western blot, and an overlay showing the coverage of that ELISA. And in this particular example, the coverage is pretty high. But it's just worth keeping in mind that ELISAs will miss certain HCPs. And in particular, keep in mind that if you're using a commercial HCP ELISA kit, it may have been developed using a different antigen. So If it was HEC-293, for example, that would have been prepared separately at the vendor, but it may be different than the HEC-293 exact, lot that is being used in another application. So there's a bit of a risk there where an ELISA could miss a significant amount of HCP. And so that's why we were just introducing the idea of using also a mass spectrometry, and that's shown on the left-hand side, a little graphic of a mass spectrometer and a LCMS trace there, and just using mass spectrometry as a complementary approach to assessing HCP risk. So the advantage there is that HCPs, if they are present as impurities at various points in the process development or even the final drug product, those HCPs can actually be identified as specific proteins. And this contrasts with ELISA, which just detects total HCP, but it may not tell you which one it is. So that's one of the advantages here of using LC-MS-MS with proteomic approaches in database searching to actually identify what those proteins are. In the middle of this slide is showing an immunoassay or immunocapture approach using magnetic beads. Again, I won't go deep into this, but this is just a way of also providing a coverage assessment using mass spectrometry, which is sort of analogous to the coverage assessment shown in the top right-hand side for an ELISA. Just going a little bit further into HCP analysis by LC-MS-MS. This slide just shows comparison of HCPs that have been discovered in different samples in the downstream process versus the drug substance. This table, like I mentioned, the advantage of mass spec, of course, is to get an actual list of what these proteins are. And then you can also get some relative abundance information for all of those proteins to compare them amongst the samples. And so of course, the abundances that are high are in red, and that's going to be earlier in the DSP, in the downstream process, so DSP1. In order to obtain this quantitative information, there are several ways of approaching it. One is using an LFQ or label-free quantification standard, different proteins that are just spiked directly into the samples. There are other quantitative ways of addressing this. And on the right-hand side of this slide, it just shows the coverage of the proteins that are actually being reported. So in order to detect these HCPs, they're digested into peptides, and it's the peptides themselves that are detected. And this is just showing some coverage of those proteins based on the peptides that were observed. Okay, and then just one last example I'll talk about, just because I was mentioning glycosylation a couple slides previously. Released glycan analysis. So a lot of the proteins that we've worked on are glycoproteins. So for example, this is a monoclonal antibody that was from a mouse cell line. One of the things to keep in mind in this case is the potential for potentially immunogenic N-glycoyl or new GC containing N-linked glycan. So if you look in the bottom trace there, the little glycan cartoon, new GC is the white diamond, which is just one form of sugar. And so the top trace is just a fluorescence trace showing all the glycans. Then there's one low abundance peak there that looked like it might contain N-glycoyl neuraminic acid. And so in order to figure that out more carefully, an MS-MS spectrum was obtained. And once this glycan is fragmented, it gives off certain signature ions. For example, that 673 ion, showing one arm of that glycan that contains a new GC, and other fragments as well. So it's just a way of checking for that type of potentially immunogenic glycan in a product. Okay, and so I think probably reaching the end of the time here. So slide 4. First of all, thank you everyone for your attention. Just to recap real briefly, we have various product-related and process-related types of molecules that might be observed as impurities, and these generally are going to be initially looked at using mass spectrometry. And so we just talked about how to apply mass spectrometry and maybe some tips and tricks as well. So I think with that, I'll stop talking and see if there are any questions.
Great, thanks, Andrew. So the first question is, how is dual detection more advantageous to inline UV and fluorescence detectors that are upstream of the mass spec?
Okay, let's see. So upstream, so yeah, UV detectors usually can be run that way. What we've seen with fluorescence detectors is the flow cells sometimes cannot handle a lot of back pressure. So just something to keep in mind, but that is a good question. It's definitely possible to run them in line in that way. The only little caveat there is with the fluorescence cells that can sometimes burst if too much pressure is put on them.
Okay. And how do you recommend looking for E. coli HCP via LCMS since they lack glycosylation?
Yeah, and so the example that I showed was just sort of one way to get some ideas about HCP present, but generally what we would do is if we're really going to conduct an HCP investigation is to start with the mock or null cell line material and characterize all those HCPs using a standard proteomics type of an approach. So once we have information about what's in that starting cell line, then those proteins can also be looked for throughout the downstream process.
Okay. Can you describe the procedures that you use to clean your machine and prevent cross-contamination and minimize biosafety risks?
Oh, that's an excellent question. What we do at Charles River is that at the end of every run sequence, we have a HPLC cleaning procedure. You know, it's designed to keep the columns clean for one, but also just to switch over the instrument from one analyst to the next. So generally, that's going to involve different mobile phases. So removing the mobile phases that you did use and then replacing them with a fully aqueous and or isopropanol, those two solutions, and running those through the instrument, possibly through the column. Often the columns will be stored in about 80% organic, so we typically will use methanol. And then once a month, we will clean the systems fully, typically using isopropanol. But yeah, different procedures may need to be applied in different instances, so those are just general comments.
And can you share any links or lists for the molecular mass change after PTMS?
Oh, for various PTMs? Yes, definitely. One thing that we look at quite often is we do have a whole we do have a whole databases that we use internally, but all of the vendor software that comes with mass spectrometers will have the option to add these PTMs to your sequences. But if you need resources that are online, one of them is ABRF Delta Mass. I've been using it so long I forget even. Apologies, ABRF. That's one place to check, but there are definitely various resources online.
Okay. And how accurate is the MAM to quantify deamidation, as it may be due to sample preparation at pH around 7 to 8 for the sake of trypsin?
Yeah, and so that's definitely one of the challenges with developing an MAM method is that significant effort has to be put into method development upfront. And for example, possibly even using a design of experiments or DOE approach. So it's not just the pH, but also the temperatures that are used and the amount of time. All of these factors can come into play. So that's why we don't typically just say, oh, every time we run a peptide map, it's going to be quantitative. No, you need to apply some effort up front to understand how to develop low artifact methods. Yes, there will always be some level of artifact, but as long as you know what that is, at least it gives you a baseline.
Okay, so it looks like we have time for just one more question. If you still have questions, go ahead and type them in and we will pass them on to Andrew directly. And if we didn't get to your question, we'll be doing the same. So the last question for the presentation is, what are some major differences between ELISA and mass spec for HTP determination?
Oh, good question. This has been a bit of a challenging area, I think, over the years, because they are so different. They've been called orthogonal or complementary. So ELISA is very quick to apply, obviously, and that's why it's very, very useful for like a QC or a rapid, maybe even a plate-based type of a method, so you can get your answers very quickly. The ELISA reagents themselves, so significant effort needs to be put into developing those reagents. So kit-based methods, again, generic type methods are okay to use for, say, phase 1 and 2 type programs, but because they don't offer that specificity towards your particular expression system, those product-specific ELISAs need to be developed using inoculations of animals independently, and that process can take 6 months to 12 months. So that's one of major differences there, is that the process to just develop the ELISA can be fairly long, but once it's in your hand, it's fairly quick and easy to use. The ELISA itself, as I mentioned, doesn't tell you which proteins are actually present. And having some knowledge about that can be quite useful if it's incorporated early enough in your program. And that's where mass spectrometry really comes into play, is that it can identify what those proteins are and then be able to monitor them to make sure also that your ELISA is actually catching those specific proteins.
Okay, great. Thanks, Andrew.
Thank you. Thanks, everyone, for your attendance.
And the recorded version of this webcast will be made available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.